Last time I was trying to explain you, and I was trying that I teach you that the philosophy behind this thing, that what is the primary stuff, what is the actual secret behind the capacity of cells, that how the different cells, they are able to execute different things. What is the difference between the cells at a cellular level, that they cells can act as a stem cell, or they can act as a differentiated version of cells. But you should also remember that the being a stem cell is not fascinating thing. I mean, being a stem cell means that you are not able to execute any function. I mean, agar main kahun, if I can relate that stuff, for me, they are functionless type of things. They're not doing any function. Their job is to carry all the potential of the cells to the next stage. And eventually, they converted themselves into all of these different type of differentiated versions of the cells. Also, I want you to understand that this is not a single step process. It's a gradual process of converting from one cell into another, and then into another, and then into enter into two different tracks, and then into another, then move into different tracks, and then into another, then into another, then into. So we eventually started the journey from the original cell, the most famous of the zygote. But we, at the end of the story, we started receiving large number of differentiated version of cells. The differentiated cells like these, they have the capacity to execute certain jobs, but they are not directly originated in one single transformation. It's a series of events. It's a series of events which gradually transform the one cell into another version. And what I can tell you is that there are different points, there are different stages where cells enter or cells experience a situation like the cell has two options to proceed from here on. Either the cell will choose to go on this path, or the cell will choose to go on this path. Once the cell is committed to start its journey on this track, those cells will never be able to produce any of those type of differentiated cells. Now they are far, far away. Their journey is different. They will be converted into this one. They will be produced these type of cells. So it really depends on the nature of cells at that time and the level of commitment they are having. Now those cells, this cell is supposed to be called as the committed cell. They are committed ones, that they are now going to produce only these type of cells. They are committed precursor cells. They will never be able to produce those things. Let me take your thoughts more on this thing. This is very important concept. I will redrawing that stuff just to make things more easily understandable for you. So we start the things from the zygote, and as I told you, of course the zygote will going to produce large number of cells, but the new cells are different, and the different cells, they will become different, and there was a time when they will become more different from each other, and they separate their paths, and they eventually ended up into different number of cells. This end, if I draw the line from here to here, this is the starting point, or I must say, I'm sorry about this. So this is the starting point, and this is like the journey towards time. If I scale this thing up like this, When the time is 0, things started. The stemness is maximum. The stemness is maximum. If I rate the stemness as 0, so the stemness here will be like, stemness here at 100, the stemness will reduce to like 0 at the end of the journey. If I give some theoretical points, hypothetical points to the differentiation, the points to differentiation here are 0, but that increases over the period of time, and the cells which are present here, they have the highest values of differentiation. They can do defined functions. Think about this one. This is the cell which is still able to execute, which is still able to differentiate into almost every other type of cells. Let's suppose this is type 1, this is type 2, this is type 3, this is type 4, 5, 6, 7, 8, 9, and let's suppose all these are different types of cells. So this is the stem cell, which is carrying that much potential of stemness that it is still able to produce all of those types of cells. What about this one? Think about this one. Now, this is the stem cell, which is capable of producing only the type 1 and type 2. And this stem cell is not able to produce any of these type of cells because that cell has changed the course of its journey from here. It can produce 1, 2, or it can produce this one. Let's name it as 1A. So it can produce all of these 3 differentiated versions of cells. So this is what we call as the committed precursor cell. This is what we call as the committed cell or the committed precursor cells. For example, hematopoietic stem cell. Hematopoietic stem cells. Hematopoietic stem cells are the ones which are able to generate all the types, all the cells which are present in our blood system, in our immune system. The cells which are present in the blood, white blood cells, red blood cells, platelets, different types of cells which are constituting our immune system, they came from the hematopoietic stem cells. So hematopoietic stem cell, if this is an hematopoietic stem cell, it will be able to give you all of those cells which can constitute the blood, but they will never be able to produce the cells which are doing other things. Let's suppose the cell number 4 is a skin cell. It's a skin cell. So that skin cell is actually, the commitment was made somewhere here. So this step changes the course of the history of the cells. They produce this committed precursor cells. Those committed precursor cells produce this one, and then they produce this one, and then they produce this one, and the story continues. So once the cells choose that direction, it enters here. It might be able to produce the skin cell. It might be able to produce the neurons. For example, if number 5 is the neuron, number 6 is the kidney cells, number 7 is the muscles. This journey will going to give rise to the skin cells, but you will never met with the cells of the immune system in this journey. They will never be able to differentiate into the blood cells, because the journey of the blood cell, the path of the blood cell is separated from here. So those cells, look at those. If you ask me, sir, the stemness is there, the answer is yes, the stemness. But that stemness is limited. The stemness present in these cells is limited. The stemness present here is wonderful, here maximum. So the stemness of those cells is limited. Those cells are able to produce large number of cells, but they are not able to produce every type of cell. Such type of stem cells are called as adult stem cells. They are called as adult stem cells. The other stem cells like this one or like this one or this, the other stem cells which has the potential to produce all types of cells, they are the ones which are called as embryonic stem cells or traditionally stem cells. Embryonic stem cells or traditionally the stem cells. So when you proceed in time, when you go forward in time, you will met with more differentiated cells and you will lose the potential to produce more number of cells. If you ask me, please compare the stemness of this one, this cell with this cell. You can see this cell is able to produce 1, 2, 3, 4, 5 types of cells. But this cell is only able to produce cell type 1 and 2. So the level of stemness decreases with the period of time. So we are harvesting the potential of those stem cells from there. I can tell you that the nature has placed different adult stem cells in our body just to utilize these things later in our lives. Like we need a constant supply of blood cells, white blood cells, red blood cells. So nature has placed hematopoietic stem cells right within our bone marrow. And they are giving rise to all these different types of cells. The tail of the lizard grow because of the presence of an adult stem cell over there. Because the adult stem cell will going to generate all those cells which are required to grow the thing or the place into a tail. They got activated when the tail is lost. But they will never be able to produce the head. They will never be able to produce the limbs because those stem cells are adult stem cells and they are defined only, their job only is to produce the tail. So if I ask you that if I take a skin cell from here, and if I know how the zygote is converted into this one, how this thing is converted into this thing, जो मैंने आपको last time initial lecture में बताया, how genes, patterns of expression of genes changes over the period of time. If we understand the secret behind this transformation, we might be able to reverse that thing. We might be able to convert the skin cell into this one, and then into this one, then into this one, then into this one, and then into this one. We might be able to convert the skin cell back into a stem cell. And that's what I can tell you. So skin cell exists here. The stem cell, embryonic stem cell exists here. We might be able to convert the skin cell back into the stem cell. And if it is possible, then the stem cell once again can be converted into another type of cell. It can be converted into the neuron, or it can be converted into another baby. So from the skin, we can put the things and we can send it back. And when it was discovered by Yamanaka at that time, it is called induced pluripotency, or converting the differentiated cell back into the stem cell. And the article which was written at that time, many articles were written at that time. They are using the phrase going back in time. And I created a video where I said it's a biological time travelers. Now you might be able to understand how it is a time travel, because the skin exists in the future. This is the future. Stem cell is the past. We all have a common past, you know. And what is our past? The zygote. Skin and differentiated cells are the future. So if we able to convert the skin cell back into the stem cell, that means we might be able to send the things back into the future, back into the past. So like biological time travel. We can convert the skin cell into the stem cell, and then we can use that stem cell to do wonderful things. So that's, I think, covers most of the biological and technical aspect of my talk. So let's check the slides now. So the life is a race, you might know, and it starts from the single cell called zygote, converted into multicells. I want you to understand that from the initial stages, if you can recall the conversion of the zygote, the initial stages of the zygote, the initial divisions, I highly suggest you to see the video of that part. You will see that cell is like converted into 2 cells like this one, and then it will be converted into 4 cells, like something was like this. You can recall this thing, and then you might see that large number of cells are produced. The important thing which I want you to ponder here, that there is no increase in the size of the overall cell structure. The overall cellular mass stays constant. The cell mass, or I must say, क्या term इस्तेमाल करनी चाहिए? Protoplast. The protoplast stays constant. So the zygote is a bigger cell. The 2 daughter cells are the smaller ones. The 4 are further smaller and smaller. So we will get smaller cells and smaller cells and smaller cells. Can anybody tell me the reason why? If you like to speak. अगर आप इर्द गिर्द हैं, मेरे पास ही हैं, जाग रहे हैं. If you like to, nobody wants to talk. G.
Sir, maybe because to make space in the uterus.
Okay, shabash. Anybody else?
Sir, they are formed from the division of the same cells, so they are much smaller.
Okay, good.
Size.
Okay, so that's a point that at this time, the zygote is not connected with the mom's body. So there is no additional food or additional resources are coming right into the system. So whatever the thing zygote is carrying, that is the only thing which is defining whatever going on here in the earlier stages. So all the material and mass present in the zygote is the only material and mass of the life out there. So they become smaller, smaller, and smaller. Once they started establishing the connection, you will see the cell will increase in their sizes and they will develop the layers and all the other things. The next phase, okay? And then eventually the baby. So this is a stage which is quite interesting.
Sir, could you explain again why it's getting smaller?
Because the zygote did not establish a connection with the mother. The system, whatever the resources the system is carrying, the system will have to depend on those resources. So zygote is bigger in size. It consumes all the resources to make 2 cells. So the daughter 2 cells, the protoplasm is same, but the cells become smaller and then become smaller and then become smaller because nothing is coming into the system from the outside until unless the connection is made with the mother. So after like 2 or 3 days, once the zygote, developing zygote attaches itself with the mother's uterus, what happens after that? It started getting the nourishment and signals from the mom's body, and then it starts growing and receiving nutrition will help them to grow and develop new structures. And those new structures are, one of them is the outermost layer to protect the cells, somehow the inner cells, and these things are called as the inner cell mass. Each one of the inner cell mass has the potential to become a baby. For example, if there are 200 cells are out there and you separate these 200 cells and put these 200 cells into 2 200 different surrogate moms, each one of them will be able to develop into a baby. We will be able to get 200 different clone babies. So what I want to tell you here is that the cells of this stage, it's like an early blastula, the inner cell mass, they are all have the potential to become everything. If you can recall the earlier slide, you might be able to figure this thing up. That this is the stem cell which is still able to produce everything. It's like more or less like an inner cell mass. So this is able to produce everything. So they have like the stem cell. More accurately, they are called as embryonic stem cells. Embryonic stem cells, most potent, huge potential to produce everything. And what we know is that they are like eventually convert and differentiate into 3 layers and then eventually transform themselves into a baby. But each one of the cell is able to produce everything at this stage. So people have harvested those cells, and when they came to know about the potentials, I mean, it was amazing. It actually started a new dimensions of life sciences. So a stem cell is actually doing 2 things. The first is that the job of the stem cell is to keep the stemness alive. How a stem cell is able to survive as a stem cell? As long as if it is keeping the pattern of expression. The pattern of expression which keeps a cell to maintain its stemness. As long as it is maintaining that pattern of expression, the stem cell will stay as a stem cell. A stem cell is producing identical stem cells to maintain the number, and at the same time, the stem cell is differentiating to produce the differentiating cells or specialized cells. In laboratory, when people are working with the stem cells, most of their energy is consumed, and I wish if you guys are working on these things, most of your energy will be consumed in keeping the stem cells in this state. You will work hard that your stem cells should not go for the differentiation. Otherwise, they will be doomed. So on every day, you want your stem cells to stay as stem cells so that you can experiment with them and you can try with them. And most of the experiments were lost because the stem cell scientists, they failed to keep the conditions. They are so delicate. The stem cells are like ladle bacche. They are like ziddi babies. So one of the conditions are gone wrong. The stem cell will start differentiating. And on the next day, the scientists came to know, oh my God, my stem cell is now a neuron. My stem cell is now looking like a skin cell, an epithelial cell. So that is the potential present in the blastocyst or in the inner cell mass. The real thing is that inner cell mass present in the blast. If you take those cells out and you give them different conditions, you can produce different types of specialized cells. This is your task to find that what type of conditions should be provided to produce the neurons.
सर ये जो अभी आप
जी
सर ये स्लाइड पीछे करना है कि नहीं? सर ये जो प्रीवियस स्लाइड थी वो वाली सर, इसमें जो है स्टेम सेल है, यहाँ पे कह रहे हैं कि सेल्फ रिन्यूअल कर रहा है, कॉपी कर रहा है, अपने आइडेंटिकल स्टेम सेल्स बना रहा है, और ये फिर आगे डिफरेंशिएट सेल्स नहीं बना रहा, स्पेशलाइज्ड सेल्स नहीं बना रहा फर्स्ट वाला.
नहीं, a stem cell is doing 2 things. The first thing is the stem cell is required to keep its own number. For example, if this stem cell is required to produce 50, is required to produce 100 cell types. अगर इसने 100 सेल टाइप्स प्रोड्यूस करनी है तो सबसे ज्यादा जरूरी बात तो ये है ना कि ये अपने आप को बचा के रखे. The first thing is that it should be able to renew itself. So first thing, the stem cell should be able to produce the stem cells so that one of them is utilized to produce the specialized cell. Then this will going to produce stem cells, and one of the stem cell is used to produce what? Is to produce the specialized cell. Then this one will going to produce the stem cells, and one of them can be used to produce differentiated cell. So अगर हमारे पास दुनिया में एक ही stem cell है और उसी को हम differentiated product में convert कर लेंगे, so we might not be able to produce other things like hematopoietic. Okay, sorry, it's from my side. Let me share the screen once again. Okay, guys, I'm sorry, it's disconnected. So first thing is that the hematopoietic stem cells, for example, I was sharing an example with you guys. In case of hematopoietic stem cell, the first job, the hematopoietic stem cell present in the bones. So the hematopoietic stem cells are present in the bones. So the first job for the hematopoietic stem cell, if they are present in the bones, is to keep renewing itself, because they need to produce a lot of differentiated cells. So they need to keep on renewing itself. But that does not mean that this thing is not. So when they renew, they produce large number of cells. Some of the cells is utilized to produce differentiated cells. But this is important. If it is not there, we will not be able to get the other type of cells. Hope this answered the question. Okay. You can speak more if you are not getting this thing. So a stem cell, I'm sure you guys are with me. Okay, Abir has raised. जी Abir, आपने कुछ बात पूछनी है तो kindly पूछिए.
Sir, I still don't get what's physically different in them. Like they have the ability to divide and differentiate and all of that, but what is it that's different in them that if we change in normal cells they can turn into stem cells?
Okay. So that is the thing which I am sharing on this slide, that if a stem cell is put, as I explained, that if a researcher want that a stem cell should stay and divide and produce the stem cells only. That's the first thing. I'm trying to elaborate your question, okay? The stem cell should divide and produce the stem cell only. Or another researcher wants the stem cell should differentiate. Remember, there's a difference between these. Cell division and what is here? It should differentiate and produce a specialized cell or produce a neuron. So there must be a difference in the conditions of these two processes. Am I right? क्या मैंने आपका सवाल ठीक से प्लेस किया? ओके, क्या ये क्वेश्चन है? क्या आप मेरी बात सुन पा रहे हैं? क्या आप मुझे कंफर्म कर सकते हैं कि मैंने आपका सवाल ठीक से फ्रेज किया है?
Yes, sir. सही है.
Okay, so a stem cell will going to renew and produce the two stem cells. That's the one situation. And in another situation, a stem cell is going to differentiate to produce neuron. So the question was that how it happens. The point is that whether the stem cell will going to produce the stem cells or a stem cell is going to produce a neuron, it depends on the condition the system is giving to the cells. So if I have like collection of stem cells, they wants to divide and produce a stem cell. That's the one thing. And that typically we believe about the cells, that the cell produce, the daughter cells are similar or identical towards the parent cells. But what about the condition and circumstances? If the conditions and circumstances of this region is different, if the conditions and circumstances and the chemicals and the transmitters of this place are different, what will happen? Now this stem cell which enters into a new region or new place where chemicals are different, they will never be able to divide and produce the stem cell again, because these conditions will push the stem cell to differentiate and produce the specialized cell or to produce the neuron. Hope this answers the question. And this is the exact thing I want you to do, that when you take out the embryonic stem cell, the embryonic stem cell are pluripotent. इसको pluripotent भी कहते हैं, इसको totipotent भी कहते हैं. It's like a strong word. that they are potential, they have the potential to produce everything. So now they have the potential to produce everything. If I ask you, can they produce the neuron? The answer is yes. Can you convert them into cardiac cell? The answer is yes. Can you convert them into the blood cell? The answer is yes. And one question which was not written here, you can ask that question to yourself, that can you convert these stem cells into the stem cells? ठीक हो गया ना? Can you keep these stem cells as stem cells? This is another condition. So if you put the conditions, let's name this condition as A. If you use the conditions A, the stem cells will stay as embryonic stem cells. And those people who want to grow the stem cell, they are working very hard to keep those conditions. If you change the conditions like B, they will differentiate into the blood cells. If you change the conditions to C, they will transform themselves into the cardiac muscle cells. If you change the conditions like D, they will convert them into the neurons. Now, what these conditions are doing? These conditions are switching certain genes and turning off certain other genes, changing the patterns of expression of the genes. It's like using and creating different tunes out of the same DNA and producing. But if you are not using B, C, and D and using the condition A, that will not allow the stem cells to change or differentiate. They will push the stem cell to stay as a stem cell. And you can use it for further experimentation and research. Ji Aisha.
Sir, can you give me an example that what kind of conditions do we change?
No, I don't.
What kind of drugs?
I'm not giving you this an example. I am asking this question to you. Okay? जब ये slide शुरू हुई थी, तो मैंने कहा था this is your task to find out that what sort of conditions I need to maintain if I want an embryonic stem cell to be converted into the neuron. मेरा सवाल है आपसे, ठीक है? ये आपने ढूंढना है. But I told you the secret. And what is the secret? The secret is that if this is a stem cell, a chemical, a transmitter, a material, a hormone, a protein, a growth factor, somebody is there which enters into the cell, and eventually it changes the patterns of expression of the genes, which slowly transform this thing into a neuron. It produces the tail and the cell body and convert them into the neuron. So what that thing is, for the people who want to do research in the stem cell, they need to know, they are expert in playing the same things. So that is something. So that was the thought when the stem cell was originally discovered, when the concept was there, that scientists were interested to harvest the potential present in the embryonic stem cells, because they believe at that time that once we have the stem cell or embryonic stem cells, we might be able to produce all possible types of tissues out of it. For example, if a patient's heart is not working well, the heart muscles are gone, we might be able to produce fresh heart muscles for the patients out of the embryonic stem cells. And we might be able to produce like the correct compositions of red blood cells for the patient as well. So huge potential was there in the embryonic stem cells. But there was a trouble in doing research with embryonic stem cells. Although the potential was huge, that one stem cell is not only able to produce the self-renewal, the stem cell, but it also giving rise to differentiated cells. So that is the answer of the question. That the one cell is not only producing the stem cell, but also giving rise to the differentiated version or specialized cells. So that is how we will be able to get a lot of cells. But from where we can get the stem cells? We can get the stem cells from the embryo. And that is the point where most of the bucks stops here. We can find them from the embryo, from the inner cell mass. Now think about the inner cell mass. Inner cell mass, or the blastocyst, or the cells from the inner cell mass. This thing comes from where? Kahan se aaya ye? These things come from number one, the sperm, which is like it's a very cheap commodity. The other thing is it comes from the egg, because they produce in millions. They produce in millions. But this is like one egg per woman per month. Normally, I mean, look at the thing, the difference, millions every day, but one egg per month in the lady. So this is like quite precious commodity. If somebody is working or doing research on the embryonic stem cells, he needs to produce this thing. He needs to get this thing. He needs to have like embryonic stem cells. He needs to develop a zygote. He needs to merge the sperm with the egg, allow them to become zygote, and then do something with this zygote so that it can develop or convert it into a blastocyst. The best place to do this reaction is the mother's womb. Alternatively, you can do the same thing in the laboratory. You can do the same things in the laboratory, and you can raise the zygote to the blastocyst level. You can raise the zygote to the blastocyst level. But once you have the blastocyst, you are all fine, because you have a lot of stem cells, embryonic stem cells, and you can use those embryonic stem cells to do research. But think what I am telling you. The precious commodity. What is the precious commodity? The egg is here. Think about the ethical concerns. So if you want to have embryonic stem cells, you need a lady, and you need the consent of using the egg of that lady, which is like once in a month, and you will going to create a zygote in vitro, and you will going to grow the zygote. Guys, please don't write your name. Sorry, it's not the time yet. So you need to convert that. Actually, I need to go back to the chat to see that koi question to nahi hai. I need to go back again and again. That's, and I'm sure I have explained this thing before. Okay, so let's go back. So the thing which you need to understand, in order to get the embryonic stem cells, the first commodity is the sperms, which are like in millions, so no problem. How about the egg? Human egg, they're very precious commodity. And the next ethical concern is here, that once you raise a zygote in vitro, what are the rights of that zygote? I mean, you are actually killing a potential human life to produce the embryonic stem cells. So that was the debate which actually haunted the stem cell research a lot. People were fighting and wondering whether they should do embryonic stem cell research or not, because they need, like, they need an egg, although they will be able to produce a lot of stem cells, and all of those stem cells will eventually be used, can be used to produce, to raise whatever they want to. So once you have this blastocyst, you get those cells, you treat those cells in different conditions to produce all those possible differentiated cells. Please do study that what are those conditions. So in the early blastocyst, and that is a very critical thing you should understand, that when you go in time, the concept of differentiation take over the things. And I don't want to share this slide today, but since somebody asked the question and the answer is here, in the early blastula, the OCT4 is the primary player. It is the primary player which is activating the GATA6, and that actually keeping the situation in this form. In the mid blastocyst state, the OCT4 is blocking the CDX2 and it is activating the FGF4. These are the primary master regulators in those conditions, which I ask you guys to study. Okay, मैंने आपको दिखा दिया गलती से, but you guys are supposed to investigate and find out the background reasons of this transformation. And in the late blastocyst, the inner cell mass, the cells are carrying OCT4, they are carrying some early markers, and they are carrying also some committed markers, some early markers, or I must say they are like autonomous environmental support markers, which allow the cells to exist in this stage for a certain period of time, like 100 cells, but each of those 100 cells are able to produce a baby. E4.5 means This is the embryonic stage of about 4.5 days. When we have like 100 cells, and those 100 cells are the very initially when people were working on stem cells, you know, they are giving other people money to do what? To donate their you know, early embryos to harvest the inner cell masses so that they can execute their research. It seems quite unethical and weird, but we have a solution now. So at the start, it was like quite weird to get the inner cell mass or to get the embryonic stem cells for doing research. So a lot of criticism was there at that time. And that lead to another type of research. So हमने sperm borrow किया from one donor, and हमने egg borrow किया. Sperm comparatively smaller होता है, इससे भी छोटा होता है. हमने egg borrow किया from another person, and हमने in vitro इन दोनों को कोशिश किया कि they should merge to make a zygote. Now अब हमने zygote का करना क्या है? The zygote will not going to produce the blastocyst until unless it develops a certain conditions. या तो आप वो conditions laboratory में पैदा करें, या आप क्या करें कि आप उसके लिए एक surrogate mom यानी कि surrogate womb का इस्तेमाल करें, surrogate womb का इस्तेमाल करें. So the first thing is that the scientists tried to use the same thing, allow the zygote to grow into the laboratory. We are fine in growing into like multi-cell stage, and that multi-cell stage is also able to give us like 4, 8 or 16 stem cells, and we can use those 16 stem cells to execute our research. So initially to ye jawab aaya. Then scientists believe that until at the stage of blastocyst, jaise hum padh rahe hain ki blastocyst ki stage tak stemness hoti hai, so why not we should produce like 100 cells ताकि हम उनसे ज़्यादा आराम से comfortably experiment कर सकें. So for that you need like more specialized and advanced condition, and also you need a womb. Now that's the point where Zunaira was asking the question, I believe, that when we place a zygote which we have created from another people, from another donors, how the receiver's body will going to take this zygote, whether they are taking this zygote as normal or not. And I can tell you that we push the receiver's body. We push the receiver's body to allow that zygote to stay. This called surrogacy, because this was not produced naturally. The hormonal actions were not there. So we give like injections, and जैसे हम किसी organ transplant में we decreases the immune system, suppress the immune system, so the external organ is accommodated. In the same way, we push the receiver's body to accommodate the external zygote as well. ये experimental procedure or part of the plan था. But इसमें humans involved थे, so human embryonic stem cells पे काम नहीं होता था. तो animals पे तो हम बेफिक्र होके करते रहे. We don't care about animals. I have another question. Ask this question. The question was that कि क्या stem cell की मदद से हम spinal cord injury को ठीक कर सकते हैं? The answer is yes. हम देखेंगे आने वाले दिनों में, ठीक है? यानी कि next class में जब हम इसके applications पढ़ रहे होंगे, this is one of them. And I'm happy you raised this point, and this truly a right sense to think कि यार अगर हम ऐसा stem cell inject कर दें जो neurons बना के connections को restore कर दे, वो might be able to restore the function. The story is not that simple, but it is possible. ठीक है?